Good morning, student. I am Pradip Bawane, Assistant Professor, Sri Vileparle Kelvani Mandas Institute of Pharmacy. Today we are going to perform very important practical of a microbiology, that is to perform a gram staining for given culture of organism, for which we need 24-hour bacterial culture. Okay. With the help of a sterile loop, we have to first prepare a smear. Okay. And this smear has to be heat fixed by rapidly rotating over the flame. Okay. And after the heat fixing of a smear, what we have to do, we have to pour 4 to 5 drops of freshly prepared crystal violet and allow it to react with the smear for about 90 seconds. Then afterwards, we have to wash the slide gently. Okay. And afterwards, we have to add grams iodine. This grams iodine, which will be act as a mordant. Okay, and this grams iodine, we have to allow it to react for 120 seconds, which will allow to penetrate the dye inside the microorganism. Then we have to move to the next step, that is also washing. For a few seconds, a gentle washing has to be given to the smear, and then we have to apply the smear with a decolorizer. Either we can use alcohol or we can use acetone as a decolorizer. Allow it to react for about 60 seconds. Okay. Again, you have to give wash, and finally you have to add counter stain, that is saffronin, and allow it to react for approximately 120 seconds. After completion of this procedure, we have to allow it to dry, and after drying the smear, put single drop of a cedarwood oil and observe your slide under 100 X. Okay. The observation you will get, either the microorganism which will be appear as a violet if it is a gram positive, and it may be appear as a red if it is a gram negative, because the gram positive bacteria which may retains the primary stain, and gram negative bacteria which will retains the counter stain, that is saffronin. The examples of gram positive bacteria and gram negative bacteria. The example of a gram positive bacteria is Staphylococcus aureus, Bacillus subtilis. Then gram negative bacteria, common examples are E. coli and Salmonella typhi. Now, the principle for gram staining will be explained by our third semester student Rachita and Hiran.
Gram staining is a differential staining technique which is used to distinguish bacteria between gram positive and gram negative bacteria. To perform gram staining, we required following reagents and stains.
This is the first reagent used. It is crystal violet. Crystal violet is a primary stain used, which is retained by the gram positive bacterial cell.
This is grams iodine, which is used as a mordant. It is used as a reagent to fix a dye.
The third reagent used is alcohol. It is used as a decolorizer, and it also removes the stain crystal violet, as gram negative bacterial cell wall contains thin layer of peptidoglycan.
This is saffronin, which is used as a counter stain, which will help to retain the dye on gram negative bacteria.
The first procedure to be continued is we have to sterilize the inoculating loop by incineration. We'll heat it up to the loop becomes red hot. Now we have to cool the loop and insert it into the media.
What is
The sample is to be placed on the slide. We have to spread the smear on the slide. sterilize the loop again.
Now heat fix the slide smear by rotating the slide over the flame. Now after heat fixing the slides, we have to add crystal violet reagent. It acts as a primary stain. 2 drops of crystal violet are to be added. We have to wait for simultaneous 2 minutes so that the crystal violet stain has been retained on the gram positive as well as negative bacterial cell wall. Now next is grams iodine. It acts as a mordant and it helps to penetrate the cell wall of gram positive. We are adding 2 drops of grams iodine and wait for 2 minutes. Wait करना है ना. After adding grams iodine, now we will wash the slides with distilled water for waiting after 2 minutes. Required amount of water has been added and the water is drained off. Now after waiting for sufficient amount of time, we will add the decolorizer, which may be alcohol or acetone. Both may be used simultaneously. The decolorizer helps in washing off the gram iodine crystal violet complex from the gram negative bacterial cell wall, and the stain is retained on the gram positive bacterial cell wall because of presence of thick peptidoglycan layer. Now we have to wait for 2 minutes. After 1 to 2 minutes, we will wash off the gram decolorizer. And then we add saffron in. It is a counter stain and acts as a secondary. And wait for 2 to 3 minutes. Now saffronin dye will be retained by gram negative bacteria. Now we will wash out the excess stain by using distilled water. After washing off the excess of stain, now the slides are kept for air drying. Now after waiting for 2 to 3 minutes of air dry, the results would be observed under microscope. The gram negative bacterial cell wall consists of thin peptidoglycan layer which has low amount of lipid, and it is dissolved by the decolorizer used, that is alcohol or acetone. Now the crystal violet and iodine complex is formed by addition of grams iodine, which would be washed off due to the dissolving of lipids present, and the gram negative bacteria may appear as pink or red under observation as saffronin is added. The cell wall of gram positive bacteria contains thick peptidoglycan layer, so it retains the crystal violet on the cell wall, and further addition of acetone or alcohol or saffronin, it doesn't have any impact on it. So they will appear as purple or blue in color. So guys, we hope that you have understood the basic understanding of gram staining procedure.
And if you like this video, do like, share, subscribe, and for more such microbiology concepts to be clear to you, just press the bell icon.
After this, one animation is played by our teacher, Dr. Nitin Nema sir, Assistant Professor of Pharmacology Department from our institute.
Thank you.
Thank you.